Okay, good afternoon, everyone. Welcome to the second session of the 13th Annual Scientific Conference of PSDB in cooperation with Macare Medicals Incorporated. Our session for today would not be possible without the support of our dear sponsors, Merck, Phil Anaserv Incorporated, and MedTest. For this particular session, we will have two speakers who would be sharing with us their expertise on rafflesia anatomy and development. We also have Dr. Jocelyn Humawan as our Q&A facilitator, and yours truly, Joanne Awingan, as your session moderator for today. We would also like to announce that we have an ongoing photo contest. Thus, we are inviting you, our participants, to join our photo contest by taking a photo of you while watching our session. You may post your photo on Twitter or FB page using the hashtag PSDB13 and hashtag PSDB13ASC. Then tag us at PhilSocDevBio rather. Exciting prizes await the winners. Okay, so join na kayo. No? Okay. Also, before we begin with our webinar proper, we just have some reminders for everyone. Okay, some session rules. First, please turn off your audio and video during the entire webinar, especially during the talk of our speakers. And while for the open forum, you may type in your questions in the Slido. You may click the Slido link, which will be given in our chat box. Okay. Please indicate your name, your affiliation, and the name of the speaker to whom your question is addressed. Okay. And for the PSDB members, please don't forget to answer the link for the evaluation form. No? It will also be given later in our chat box. Okay, so I hope everyone will follow these simple rules to ensure that we will have a focused and orderly webinar for today. And lastly, before we begin with the webinar proper, okay, for those who are asking about how to get a certificate, here are our requirements on how to get one. First, you must be an official member of PSDB, registered in an annual scientific conference. Second, make sure to submit at least one evaluation form in the evening session and also on the Saturday session. Please do take note that the evaluation forms will open at the end of the session until 11:59 in the evening only. Okay, so I hope you will be reminded of this, participants. Okay. Okay, so at this point, I think we are all excited now to hear from our speaker. So let us begin. Our first speaker for today obtained her PhD in biology and is currently an instructor at the Institute of Biology, University of the Philippines in Diliman. She also obtained her master's and bachelor's degree in the same institute, finishing the latter as cum laude. She was a former vice president of the same society, PSDB. I think that was in 2015 to 2016, if I'm not mistaken. Okay, and to share with us her research on the development of Rafflesia Consuelo Way within its Tetrasigma host and the patterns in morphology and distribution of the Philippine Rafflesia, let us all welcome Dr. Erica Marie A. Bascos. Welcome, Dr.
Hello.
Okay, so I will stop sharing now so you can share your screen.
Okay.
Okay.
Right. So can you see my screen now?
Yes, we can see it.
All right, so good afternoon, everyone. I'm here to talk about one of the most extraordinary plant species you can encounter, and that is, of course, Rafflesia. So, since its discovery in the year 1818, Rafflesia has been considered as the greatest prodigy of the plant kingdom. And as I explain what we have found about how it initially develops and grows, you will surely see just how unique and amazing this plant really is. So today, I will be presenting the important findings of our research, which has been recently published in the journal Planta. And it is all about the early stages of development of the Philippine endemic Rafflesia consueloe within its host. So whenever people hear about members of genus Rafflesia, they'd immediately think about their enormous, foul-smelling, fly-pollinated flowers. But some people may not automatically realize that this plant is actually a parasite. Specifically, it is a holoparasite, meaning it completely relies on its host for nutrition and survival. It is a plant that does not photosynthesize at any stage of its development. So the hosts to all members of Rafflesia belong to a single genus, Tetrastigma, which is a woody vine or a liana. And the Rafflesia parasites would grow on the roots and sometimes on the stems of Tetrastigma, but the most common site of Rafflesia infection are the roots, which grow profusely on the forest floor. Now, with regards to the life cycle of Rafflesia, it is clear that focus is given on the growth and development of its reproductive structures. So it usually starts with the seeds, which carry the parasite in a form that can infect the host. However, how the seed germinates and how it inserts the parasite inside the host are still unknown. So the next stage that is described is already the emergence of the flower bud after successful host infection. So the buds would grow and bloom into the large flowers, which are pollinated by flies, and the flowers may stay in bloom for several days before they start to senesce. And the female flowers that are successfully pollinated will develop into fruits. The fruit looks like this. It is actually a berry and it contains seeds that may number from several hundred thousands up to a million, depending on the species. And the fruits are reported to be eaten by rodents, wild pigs, and even ants that may act as seed dispersal agents. Now, looking at how its life cycle is usually presented, it is evident that a certain part is not discussed, and that is its vegetative development. So these are the stages that occur within the host body, completely invisible to the naked eye, and leave no external trace on the host organs prior to the emergence of the flower buds. Now we have to take note that Rafflesia is different from other angiosperm parasites in that it has no vegetative shoot. So it has no externally visible vegetative organ of any sort, no leaf, stem, or root form during its entire development. So to understand how different Rafflesia is compared to other angiosperm parasites, we can compare it with Striga. This is a well-known plant parasite which infects the roots of cereal crops like corn and rice. So in Striga, the vegetative phase can easily be observed from seed germination, organogenesis, haustorium attachment to the host root, and its vegetative organs can easily be identified. Now again, in Rafflesia, vegetative organs are not formed, so its entire vegetative growth occurs inside the host body where it exists as an endophyte. So endophyte is a term used to refer to an organism that lives inside the plant. And the Rafflesia endophyte is very small, it is microscopic, and it is said to spread invasively through the host tissues and eventually lose contact with the initial penetration site. Therefore, in order to observe this phase of development, we would need to look deep into the anatomy of the host organs where the endophyte is known to exist in a reduced form. And this is what we aim to do in this investigation. Our study species, Rafflesia consueloe, which was discovered in Pantabangan, Nueva Ecija, is the most recent addition to the list of recognized Rafflesia species in our country. So its average diameter is just 9.73 centimeters, and it is the smallest Rafflesia species in the world, and it's only found in the Philippines. So you can see in the right photo just how small the flowers are. So they can easily fit in my palm. There. So in Pantabangan, the Rafflesia consueloe flowers can be found in different stages of development all year long. And a single tetrastigma vine can serve as host to hundreds of buds. So the access to the Rafflesia consueloe populations there gave us an opportunity to perform studies that are usually difficult to conduct when there is very limited plant material and individuals to sample from. So we identified the infected vines in the area based on the presence of the reproductive structures like the buds, the flowers, the fruits, and also scars from previous flowering events. And we also observed that the tetrastigma vines, which have been previously considered free from infection, may suddenly start producing buds. So we wanted to find out if we can identify vines already serving as hosts to Rafflesia consueloe parasites, but in their pre-reproductive stage. So this would allow us to identify the host individuals that are actually infected but are not yet showing signs of external Rafflesia growth. It is also important to note that for some Rafflesia species, flower growth can be observed up to the aerial stems here. But because endophyte growth is invisible to the naked eye, we cannot tell if internal infection can reach other parts of the host prior to the emergence of the buds. So for Rafflesia consueloe, we wanted to see if internal infection can reach the other organs, like the stem, for example. So in a recent publication, a summary of the life cycle of Rafflesia consueloe was presented together with the timeline of its major life history events. So in this diagram, it is clear that the mechanisms for seed dispersal, seed germination, together with the different stages of vegetative growth still remain unknown. So with that, we conducted this study which aimed to examine the distribution and growth of Rafflesia consueloe endophyte within its host and to describe the early stages of parasite development within the host tissues. So in order to do that, we collected root and stem segments from tetrastigma vines with and without external signs of infection. So we got 3 10-centimeter segments per individual there. So we collected it at 1 meter interval per individual. So morphologically, the stems were distinguished from roots based on the presence of nodes and internodes and their slightly flattened appearance. And all the collected parts were placed in formalin acetic acid. Now, I know that there might be some concerns regarding the possible harm and damage that may be caused by tissue collection to the vine host of Rafflesia consueloe. However, based on our continuous site monitoring, there were no instances of host death months after we performed the tissue collections. So the infected vines continued to produce buds, and the vines without signs of infection were also able to continue growth, and some produced buds after several months. So for histology, we just follow the standard protocol for tissue processing, blocking, and microtome sectioning. And the stains used were the nuclear stain hematoxylin and the cytoplasmic stain eosine. And the resulting slides were viewed under a light microscope and photographed using the Dyno-Eye camera. So we're very happy that we were able to generate sections that showed the different tissues of the host very clearly. So these are the micrographs of sections obtained from the host stems. Here you can see the secondary phloem and xylem regions. So the vascular tissues flank the central pith. And outer to the cortex, you can see the cork cambium, as well as the lenticels in the periderm layer. There. So the secondary phloem region here is composed of sieve elements alternating with fibers, hence the stratified appearance. And you can see wide vessels in the secondary xylem region, and the cambial zone is here in between the xylem and the phloem area. So those of you who are teaching plant anatomy courses will surely be familiar with all of these. These next set of micrographs show the anatomy of the tetrastigma root. Again, it is very easy to identify the different tissues like the periderm, the cortex, the vascular tissues. So in the xylem region, you can see vessels with different diameters. And similar to the phloem of the stem, the phloem of the root also has sieve elements alternating with fibers, and you can see the vascular cambium in between the secondary xylem and phloem. Right. Now, for this study, we generated and analyzed a total of 1,278 root and 684 stem transverse microtome sections obtained from host vines with and without external signs of infection. And we found that 61% of the root sections showed evidence of endophyte growth. On the other hand, all of the 684 stem sections, none of them contained Rafflesia consueloi endophyte. So it's 1,962 sections. So why so many sections, you may ask? Well, we were trying to reconstruct developmental events happening inside the woody plant. Of course, it would be nice if we can do time-lapse observations of these stages, but that is just not possible in this case. And the histological sections can be likened to still photographs showing us something that had already happened. So the more photos you have, the easier it is to see the progression of events, and the easier it is to reconstruct the stages of development. And this is actually one of the things that we were able to show in our study, that the vegetative development of Rafflesia consueloi can be easily examined through histology of the host organs. So we saw that the endophyte cells are easily distinguishable from the host cells due to their larger, more prominent nuclei. So these photos show endophyte strands growing among the phloem sieve elements. So the ovoid nuclei of the Rafflesia consueloi endophyte are roughly 3 times the size of the host cell's nuclei there. So based on the average diameter. And the endophyte cells also have a remarkably different shape compared to the cells in the various tissues of the host. So the endophyte cells are more elongated or fusiform. And then we found that the endophyte of Rafflesia consueloi can be effectively visualized even without the use of special stains. So common stains like hematoxylin and eosine allowed easy identification of the endophyte cells. So in the right photo, which is from our study, you can see that the nuclei of the endophyte appear dark and distinct in the root transfer section. The photo on the left shows a Rafflesiaceae endophyte from another study using a different set of stains. There. So the endophyte of Rafflesia is actually believed to originate from the cells of the proembryo found inside the Rafflesia seed. So if you're going to compare a Rafflesia seed to a seed of a typical dicot plant, you will not expect to find a true embryo inside the Rafflesia seed. Why? Because again, Rafflesia does not give rise to a vegetative shoot. Instead, the Rafflesia seed would just contain an undifferentiated proembryo. And what we know so far is that the cytology of the endophyte cells is very similar to that of the proembryo cells, indicating that the endophyte likely originated directly from it. However, how they got inside the host, that's still a mystery. So there are several possible explanations for the large nuclei seen in the endophyte cells. It could just be a passive consequence of having unlimited access to the host resources. It could be due to host to parasite, horizontal gene transfer, or due to endoreduplication. And another possible explanation could be the loss of the housekeeping genes from the plastogenomes and their transfer into the nuclear genome, where they just become non-functional. And this was recently discovered in another plant holoparasite, Aeginata indica. So whole genome analysis of Rafflesia plants would be useful in confirming these hypotheses. And in terms of distribution, the endophyte was predominantly observed in the axial secondary phloem, the vascular cambium, and the secondary xylem region. And they were absent in the outer tissues, such as the periderm, the cortex, perivascular fibers, and the phloem ray. And the endophyte was mostly observed as rows of cells oriented radially in the transverse section. So here you can see the endophytic strands found within the phloem, and we found that majority of these uniseriate strands traverse the vascular cambium. And these photos show the uniseriate endophytic strands found within the secondary xylem. And we found that the endophyte was able to grow around vessel elements like these, positioning itself in between the lignified cells. And the endophytic strands found within the xylem have cells that are longer and thinner compared to those found in other tissues. There. And under the microscope, it was very easy to see which is an uninfected root from an extensively infected one. So here you can see on the right all of the endophytic strands within the xylem tissue. And then here they are absent. So in the root sections, we also found larger multiseriate endophyte cell clusters, though they are much fewer in number compared to the uniseriate strands. And these were mostly found within the xylem region. So they're initially forming there within the lignified tissues and pushing their way through the outer tissues in the more advanced stages. And in some of the root sections, we were also able to observe some stages of early reproductive growth. So here you can see a very young floral shoot, and its basal part can be seen to be in direct contact with the xylem. There. And of note, we only found developing floral shoots in roots that had external signs of infection. And here is a longitudinal section of a developing floral shoot. So on either side of the floral apex, you can see the bracts, which are the first floral structures to form. And the cells near the basal part of the protoform are more elongated than the cells in the apex, indicating that cell elongation occurs acropetally or from base to apex. Also, unlike the cells of the early endophyte stages, we observe that the cells of the protoform divide more rapidly. So this is evidenced by the presence of cells in various stages of mitosis. So in the early endophyte stages, there were no mitotic figures. This is indicative of very, very slow cell division. So here in the protoform stages or in the floral shoot, the cells are dividing faster. That's why we can now see these different stages of mitosis. Right, so in a recent concept paper, several hypotheses regarding endophyte growth were presented. So in the first hypothesis, the early endophyte cells develop into uniseriate strands before transitioning into the reproductive stage. In the second hypothesis, the endophyte initially starts as cell clumps prior to reproductive growth. And in the third hypothesis, the uniseriate strands develop into cell clusters and then into the primordial flower bud. Now, our results are in line with this hypothesis, that the endophyte cells become uniseriate strands, which then become cell clusters before transitioning into the reproductive protoform stage. So in our planta paper, we were able to update this current information on endophyte growth progression based on the results of our study. And our study also found that Rafflesia consueloae infection is restricted to roots. So some Philippine Rafflesia species like these four, have been reported to infect the stems of their hosts. And these reports are, of course, based on visual evidence of flower growth on the stems. And although presence of Rafflesia consueloae buds have never been observed in the aerial stems of the host, we included transverse stem sections in our study in order to look into the possibility that the endophyte growth may reach the host stem, even without external manifestation of infection. But all of the section stems had no evidence of endophyte growth. So all the tissue regions did not have endophyte cells. So based on these findings, we have reason to believe that the host stems are rarely or not at all infected by Rafflesia consueloae. However, increased sampling may be needed to verify this result. So the hardness of the host stems could be a contributing factor as to why the infection does not reach this organ. So in preparing the stem segments, we noticed that the stems of the host were very hard to cut compared to the roots. So the periderm of the roots can also be easily peeled off even by hand, which is unlike the stem periderm, which is firmly attached to the underlying tissues. And also based on the anatomy, the stem tissues were observed to have to be more dense. So we have denser tissues in the stem. So it is possible that the Rafflesia consueloe pro-embryo from the seed or the cells of the endophyte may not be able to penetrate and grow within the harder, denser tissues of the host stem. And another possible explanation is that Rafflesia consueloe seed germination may require presence of specific signals that are only produced in the host roots. The parasitic strigoplants, for example, require host-generated germination stimulants in the form of strigolactones. So in the absence of these signals, the seeds will remain dormant. And similar root-derived signals from hosts have been identified as germination stimulants in other root parasitic plants. So it is possible that Rafflesia consueloe seeds require a similar germination-inducing signal that can only be derived from specific tissues of the host, like the roots. And the lack of success of in vitro seed germination and plant tissue culture experiments on Rafflesia could be due to the absence of these host-derived signals. So, of important note, all reports of successful Rafflesia propagation experiments involve inoculation of live hosts. So this could imply that the still unknown factor required for Rafflesia seed germination may only be present in living woody tetrastigma vines. Next, our study allowed detection of Rafflesia consueloe endophyte in vines without any external manifestation of infection. So of the root section examined that had no external signs of infection, 22% were still internally infected. So on the outside, the roots of these seemingly uninfected host plants were completely devoid of buds or scars. But yeah, it's clear that root infection does not always manifest externally. So the said roots were found to exhibit one of the following patterns of endophyte distribution: single-celled or short uniseriate endophytes solely found in the cambium area, numerous uniseriate endophytes scattered within the vascular tissues, or a combination of these uniseriate and few multiseriate endophyte clusters in the vascular tissues. So all of these suggest that the Rafflesia consueloe endophyte could exist in the host root as small, slowly dividing groups of cells without immediately transitioning into the reproductive stage. And if the endophyte can indeed persist in the host root for a long time without developing into flowers, then internally infected host individuals without any signs of infection can easily be misidentified as uninfected. So in the past years, a number of Rafflesia seed inoculation and host grafting experiments were reported. And in some reports, the bud growth on the host root was observed between 2 to 7 years after placing the seeds within shallow incisions on the root. Other studies report successful grafting of the host, implying that the endophyte in infected parts have a way of spreading to uninfected rootstock. But in all of these reports, none of the host vines were checked for presence of the endophyte before carrying out these experiments. And if the endophyte cells can indeed persist in the host root for a long time without developing into flower buds, then internally infected host individuals without external manifestation of Rafflesia growth, again, can be misidentified as uninfected. Therefore, we recommend that prior to any propagation efforts to seed inoculation or grafting, the checking for the presence of the endophyte cells within the tissues must be performed. So this will help to accurately determine if the subsequent bud growth is truly a result of the inoculation or grafting procedure and not due to the pre-existing endophyte cells within the host tissues. And next, the cells of our Rafflesia endophyte must reach the vascular cambium prior to proliferation. So it has been previously speculated that the Rafflesia endophyte initially infects either the vascular cambium or the phloem prior to their spread to the other tissues. Now, in our study, we observed that sections from the roots without external signs of infection mostly had endophyte cells exclusively found within the vascular cambium. So the tissues outer and inner to the cambium did not have any endophyte cells. So this indicates that the parasite cells must reach the vascular cambium prior to proliferation and provides evidence that the vascular cambium is the initial site of infection. So the subsequent periclinal division of the fusiform initials of the cambium as they form the axial secondary xylem and the phloem likely resulted to the radial spread of the endophyte cells because they were able to divide together with the meristematic cambium cells. So this implies that the cells from the seeds of Rafflesia consueloe must be able to reach beyond the tissues of the bark in order to initiate infection in the host. And next, extensive vegetative growth did not precede reproductive development. So we found that the occurrence of a limited degree of infection, such as the one that you see here, wherein the endophyte is limited is in the vascular cambium region only, does not prevent the development of more advanced reproductive stages within the same tissue. So the section you see here was from a root that already had developing buds. But as you can see, isolated endophyte cells were seen solely in the cambium only there and nearby phloem region. So endophyte distribution in this sections is so limited compared to some of the infected roots that still did not have developing flower buds. So these suggest that extensive growth of the endophyte within the host vascular tissue is not a prerequisite to the onset of reproductive development. So floral shoot development may ensue as long as a group of endophyte cells in contact with the vascular tissue, particularly the xylem, could continue dividing to form the floral shoot. There. And then lastly, we found that host roots had varying degrees of infection. So the abundance and distribution of the endophyte in the root tissues obtained from individuals with and without external signs of infection varied widely. So the extent of infection ranges from just a few isolated cells confined in the cambium area to widespread growth of numerous filamentous and a number of multiseriate cell clusters in the entire vascular area. There could be several possible explanations for the differences in the degree of infection observed in the roots examined. The simplest explanation could be just the section with fewer endophytes growing exclusively in the cambium region were obtained from more recently infected roots. So as I've mentioned before, the vascular cambium is the hypothesized initial site of endophyte infection. So this would explain why many of the roots obtained from the host plants without externally visible signs of infection only had endophyte cells within the cambium only. Another intriguing possibility is that the roots with limited endophyte growth may have been infected for a longer time, but these host individuals may have a higher resistance to the infection. Now, this remains plausible because there were roots that already had Rafflesia consueloe buds, but the growth of the endophyte in the sections was confined to just a small area. And it is also possible that different host individuals may have varying capacities to prevent or limit the extent of infection. So this type of resistance has been demonstrated by the host of striga plants. So the parasite is unable to penetrate the endodermis of the resistant host because they can produce resistance proteins that can cause growth arrest or necrosis of the parasite cells. And then lastly, the differences in the abundance and distribution of the endophyte within the host tissues could be related to the number of seeds that were involved in the infection process. So it has been previously shown that a single tetrastigma vine can house multiple parasite genotypes, indicative of multiple infections. So roots with higher endophyte abundance may have been initially penetrated by a greater number of parasite cells from multiple seeds. Alternatively, different parasite genotypes could have varying degrees of infectiousness, hence the differences in the extent of endophyte growth. There. So let me present the conclusions of our study this way. So the cells contained in the seeds of Rafflesia consueloe must reach the root vascular cambium to initiate infection. And these endophytic cells may remain confined within the cambium area or spread radially in the surrounding tissues if they're able to divide together with the meristematic cambial initials. And the parasite may remain in the vegetative stage for long periods. Thus, host individuals without external Rafflesia growth may not be necessarily be free of infection. And the endophyte cells initially start as uniseriate strands and grow into multiseriate cell clusters within the xylem before developing into the reproductive protoform stage. And the development of the floral shoot may ensue even in the absence of widespread growth of endophytes within the host root, indicating that extensive vegetative growth is not a prerequisite to the onset of reproductive development. And then meristematic activity of the floral shoot meristem will produce the structures that we see in hosts showing external signs of Rafflesia infection. So for Philippine Rafflesia, there have been many investigations focusing on taxonomy, reproductive growth, and phylogenetics. For this simple study of ours, using Rafflesia consueloe as a model, we just asked a very basic question. What happens during vegetative growth? And we were able to give you a rare opportunity to see the developmental stages prior to the emergence of Rafflesia from its host. Also, a study like this has never been done before for any Philippine Rafflesia species. So before, only the events during the reproductive development of Rafflesia consueloe are known. So now we can describe the early stages of infection and vegetative growth within the host. So this just shows that there are still many aspects of Rafflesia biology that we can focus on, and the information that we get from such studies would give us a better understanding of how we can protect and conserve these unique plants that are only found in our country. So thank you so much, everyone.
Okay, thank you very much, Dr. Bascos. Thank you for your very interesting topic, no, for introducing us to this new species of Rafflesia, a very cute species at that, no? Ang cute nila, ang liliit talaga. Okay, so, okay, so let me share my screen now. Thank you very much again, Dr. Erica. At this point, we would like to acknowledge first the presence of Dr. Annabel Herrera, our founding member of PSDB, and also our advisor, Dr. Sonia Jacinto. Good evening, Doctors Herrera and Dr. Um, Jacinto.
Good evening.
So, it's so nice for you to attend po. Thank you very much for gracing our webinar.
Thank you rin, Joanne.
Yes, ma'am. Okay, so um, I'm pretty sure na after ng talk ni Dr. Erica Bascos, there are many questions now that are already starting to flood our Slido link. Okay, kasi I'm sure just like me, they are now interested about the new Rafflesia species that is, according to Dr. Bascos, is found in the Philippines. No, I later ko na lang itatanong if it's may question din ako later on. Anyway, so at the interest of time, we will now move on to our second speaker of the day. So we have, okay. So our second speaker is a graduate of Master of Science, major in plant breeding at the Universitas Gadjah Mada in Indonesia. He is currently taking his Doctor of Philosophy at the Department of Chemical Engineering in Abo Academy University in Finland. He published over 30 scientific articles, 3 books, and 1 book chapter. He is currently a member and associate member in Royal Society of Biology and the Linnean Society of London, respectively. His research focuses on Rafflesia developmental biology, histoanatomy, secondary metabolites, alternative sourcing, and biotechnology for the rare flora conservation. And to give his talk on Rafflesia anatomy and development, let us all welcome Mr. Adhityo Wicaksono. Let's give him a big hand.
Thank you.
Thank you, sir. I will stop sharing so you can share your slide now.
All right. I will continue. Okay, can you see my slide right now?
Yes, sir. We can see it now.
All right. Hello, good afternoon, everyone. Thank you for the opportunity to having me here in this conference. So in this afternoon tea time for all of you, for all of us, I want to share all my history with this amazing alien flower in the world. Well, the first time I see this flower is in 2012, and at the moment I become really curious about what is this plant, what is it doing, and why it doesn't have any leaf, root, or something. It's just like a flower, and then I know it when it blooms. It smells like a toilet. Okay, so this is take a seat, and I will tell you the story of like a decade long of my study with this flower. Everything is impossible without the help of my research supervisor in Bogor Botanical Garden, Ibu Sofi Mursidawati. She's the one who showed me the magical word of Rafflesia, and the one who also allowed me to do a lot of stuff and to make a brainstorm at a lot of idea for this amazing flower. Okay, let's first talk about this, the progress of the anatomy and developmental study. The thing that inspires me a lot to do this research is based from Bu Sofi Mursidawati experiment of grafting done in 2007. The specimen of Rafflesia padma itself was taken from Pangandaran coast area of West Java, Indonesia, and the tetrastigma was one from Pangandaran itself, the one with infected with Rafflesia, and for the rootstock, the tetrastigma has been grown in Bogor Botanical Garden. So the scion is from Pangandaran, and the rootstock is from Bogor Botanical Garden. She did the two methods of grafting. We have here as the cleft grafting and finger grafting in 2006 or 2007, and 2004 if I remember correctly, because it's paper in Sidawati et al. 2015. And the amazing thing happened in 2010, where the first grafted flower bloomed in Bogor Botanical Garden. Here. This is the flower of Rafflesia padma, observed under photographic time lapse. And as you can see here, the process of blooming cannot be predicted, because once it's the once of the perigone is just opening, you can tell how long it will take it to the bloom, to the full bloom at the day, but it will be happen in the same day. And after that, I'm starting to read. Okay, this is a rare flower, right? This is an endangered flower. So why nobody done any attempt to do of this biotechnology approach other than the grafting approach? Even in 2012, back then, I have no idea if this grafting approach is succeed. So, and then I read this paper from Bapak Lazarus Agus Sukamto. He managed to do the plant tissue culture using flower bud of Rafflesia arnoldi, but unfortunately, it's all failed. The same experiment also done by Ibu Sofi herself and Ibu Handini in 2009 on Rafflesia padma in Bogor Botanical Garden. The amazing thing is the tissue, flower tissue was swollen, but that's it. Nothing happened after that. And then only the craziest news that I heard in 2010, again by Pak Agus, is also on Rafflesia arnoldi. The callus, in the first time in the world, is successfully generated. People at the time felt like, okay, is that really Rafflesia, or are you sure it's that Rafflesia, or is that tetrastigma, the host? But well, who knows? because the callus was generated using the MS media and the synthetic auxin picloram. Okay, the callus is successfully generated, but no organogenesis was successful after that, because the tissue culture was induced using kinetin, the cytokinin, which is normally used for the organogenesis, but nothing has happened. Okay, and then I also doing I tried the ex tissue culture by myself in 2015. Still, nothing happened. As you can see here, in the right is my attempt, because I did the tissue culture using the activated charcoal media in attempt to alleviate the browning process by the flower. Nothing happened in just a week, but later the mold start growing. I don't know after that if the mold come from the tissue or somewhere else. And combined, now me and Bu Sophie tried again with the tissue culture. Still nothing happened. Bu Sophie herself was also trying to germinate the seed in 2015 in just a pure MS media culture, and also using the tetrastigma extract, and also co-cultivation using tetrastigma seed, but all bear no results. And then in 2017, following a year after my trip to Finland to do my first year for my PhD, I have this big question. Okay, if you can see in the picture, the grafting occurred was done in this green area, right? In this green stick area. But as you can see, there are many flower bud found in this A, B, C, and D, which indicate somehow that the tissue is probably just creeping around after following the grafting, and at that moment we have no idea how Rafflesia managed to do that. I make a discussion with Dr. Lakezar Nikolov, or I call him myself Luke. How is the possibility of this Rafflesia to form like a creeping or a liana inside the host tissue? My first idea, as you can see in the top right, is that the flower is basically grow after on the liana, growing inside the root of tetrastigma. But then Dr. Luke said to me, I don't think so. It's basically fragmented. It's a fragmented tissue found over all over the root. So I don't think it's possible. It's that Rafflesia is basically in the lianas, and we have no idea how is it look like inside the root. But all we know that the endophytes, as previously mentioned by Dr. Baskos in this presentation, and in her presentation, that there's a lot of uniseriate strands found in the root of tetrastigma infected with Rafflesia. So basically, all the sessile tissue of Rafflesia, not creeping around like a liana. But how does it manage to go and spread around the root? And then in 2018, for the first time, and after also make a consultation with Dr. Nikolov, and I said like, look, is it possible? Why did you do the longitudinal section? And then he said that it's really hard because we have to cut the root of tetrastigma along the fiber, and it'll make it the sectioning really brittle. So well, and I talked to Bu Sophie. Bu, is it possible for us to do this sectioning? Where we have we have the experienced technician in our lab, right? Okay, let's try it again. Okay, and this is what happened. For the first time, we managed to see what appears to be the endophyte of Rafflesia in the longitudinal section of the infected tetrastigma root. It appears to be a cluster. What previously said as a uniseriate strand, but we don't found it in a uniseriate strand because it will be really hard for us. Imagine that if you see a uniseriate strand like this, and you make a full sectioning like this, it will be more like this, right? It will be something flat, or it will be just something, a section like this. And it will be really, really hard to find something like this. But after we want to make sure if it's true Rafflesia, as you can see here, the size of the nucleus, as documented by Luke himself in his paper in 2014, the size of the nucleus is indeed larger than the cells around it, surrounded, and it is located in the vascular cambium of the host, which raises a new hypothesis from me. What if these endophytes basically spread along the host tissue using the power on the shear force of the cell division occurred in the vascular cambium? Imagine you're playing with the squishy, right? All you have to is there's a surrounding pressure, which will make the endophytes spread around, and during all the process, it will make in the long process, it will appear to be similar to what happened in the cancer process of metastasis, something like that. That's my hypothesis, and we have no idea if it happened like that, because as Dr. Basko said in her previous presentation, it will be really, 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 really hard for us to see what happened, because it's inside the plant tissue, and plant tissue is hard, unlike in the one inside our body, where our body is basically soft and you can see it using a CAT scan or something. So someone with cancer can be monitored easily using the recent technology. How about this one? This is something really hard to see. But then in another progress in 2019, we managed to make the first colored section of the flower histology. This is from Rafflesia Padma as well. As you can see here, it came from the perigone lobe. The lobe is something like the one like the petal, and then the one in the perigone tube that covered the spiky area, the processes, it's of the Rafflesia. The histology itself is already basically the first done by Nantavan and Zuvit from Thai, but it was done in black and white, and probably so I'm proudly present here the sectioning done in color. We use the same procedure like the one with the endophyte. We use the fast green and saffron staining to color our tissue. Amazingly, similar to the concept of the endophyte, where everything comes so minimalistically, the organ of Rafflesia itself here is very minimalistic. Why? Number one, the vascular tissue here doesn't have phloem. Well, somehow after I read through the reference, it's obvious. It has no phloem, but it's all basically just composed by xylem and the vascular parenchyma. All the tissue surrounding it is quite similar to one another. The one difference is also the epidermis. The external epidermis, or we call it the abaxial epidermis, is basically all form of flat epidermis surrounding with the flat cells, and the internal epidermis, or I call it the adaxial epidermis here, is form a papillated layer that I suggest it's also contributing to the other emitting chemicals, but we have no clues until now. In 2010, when the pandemic hits us really hard, there's one of our colleague in the community of the endangered flora of Bengkulu, where they found Rafflesia arnoldi in their aerial form. One that came to my mind is basically, okay, is that so basically it grows on the stem, right? And the problem is, if you see the anatomy of the stem, previously also mentioned by Dr. Baskos, it's really dense. It's all the vascular tissue stacked to one another to form like a very solid radial shape. While on the other hand, there's a lot of space. Also, not that much. There's a few space in the root tissue that available only on the root. So on the stem is very dense and the root is have a space a little bit. But then there is the second question: is that really is that a stem, or is that a direct root? That's my question. So then I make a brainstorm with Bu Sophie again, and we have to do something about this because everyone around the world will say, okay, this is a root, this is stem. We have to fix this, and we have to provide an idea for everyone to read our paper to make sure that this climbing vine is actually a root or stem, to settle it once and for all. So the idea is basically, imagine this is the stem, right? The stem, the liana, and this is the flower. And I propose that we make a partial sampling here, because if you see the cuttings of the stem or root in only have in this is not that much deeper area, you can easily tell that, okay, that's a root, okay, and that's a stem. Why? Because the stem has a pith, right? While the root has no pith, and there are spaces. There are gaps between the vascular tissue. And what we discovered that on our currently published paper in Journal of Plant Development, and also in yes, we did it on also in Philippine Journal of Science, that the one we did also the same experiment on Rafflesia padma in Bogor Botanical Garden. We tried to sample the erect vines, and guess what? All we found, that's not a stem, that's a root. So how is that possible? So the only possible explanation from me is basically when the vine was still in younger stage, the surrounding environment, especially the tree, is also growing up, right? So imagine this is the young tissue, and then there's a growth from the surrounding plant, and also supported by the rain caused erosion, and then the vine will become more erect and hence become aerial. So from this, somehow I hypothetically want to say that Rafflesia is basically a root parasite, not root and stem parasite, although this require further investigation. And I'm happy to see it if someone basically want to manage to prove to all of us that Rafflesia is also a stem parasite, but we need to see that, right? We need the publication and a proof, solid proof that the Rafflesia can grow up to the stem region. Because why? Because there is a transitional area, right, from the root and the stem. From the beginning of the development, stem always a cambium to support the growth of the vascular tissue. However, root only have cambium once it's entered the secondary growth, from what I learned. So what happened before that? Cambium itself is basically growth from the peridermal tissue during the transition from the primary root into the secondary growth root. So that's the clue. I don't know what happened after that. We need more study about that. Another crazy thing I did in 2020 is I injected this Rafflesia bud using plant growth regulators or hormones using auxin and cytokinin. As you can see here in the graphic, during the first 2 weeks, it appeared that both cytokinin and auxin has the impact to the tissue, and then yes, it's completely normal. It's quite similar to the control. We initially want to do it with gibberellin, but the number of the available flower buds is not supporting our study back then. But the interesting thing is, despite both of them are growing bigger, only the Rafflesia injected by auxin undergoes the transition from what it called is the cupule stage into the cupule bracts stage, which is cracking, and the bract inside is showing. So that's probably added something more since the Agus Sukamto and Mujiono study in 2010. that auxin is basically the plays significant role in Rafflesia growth. But who knew? Again, all question about everything about Rafflesia in the end will be who knew. All I can do is just making a hypothesis, doing a study, add something more. But whenever I do the study, more doors of question will be open. Now, whatever I did, since I have no longer have an access because there's a pandemic that hit us really hard, we transition into the age of bioinformatics, and everything is gone to the computer model, as you can see here. I did the simulation using the available data from other Vitaceae. The fit is the grape. How big is basically the mechanical expressed by the growing flower of Rafflesia. Whether they're growing aerially because they're vertical, they have to sustain gravity, and versus the terrestrial Rafflesia. Are there any difference? Are there specific adaptation that make the Rafflesia is different from the other cousin? And I did this modeling using finite element modeling. And well, we want to do further study from this in the future. It's in progress. Also, from time to time, Rafflesia has been recognized as a source of the ethnobotanical medicine, right? There's a claim for that Rafflesia is basically has a property of wound healing that cure the postpartum wound, and also used for aphrodisiac and something like that. So I managed to gather the data from a journal using 21 metabolites from across Rafflesia. I used the metabolites from Rafflesia and Sapria and perform some molecular docking to see if they really have that potential. In the end, only anti-cholesterol, antiviral, respiratory virus, and wound healing, antibacterial, and antifungal are confirmed. No proof for the anti-neurodegenerative against Alzheimer, no proof for the aphrodisiac. So basically, that's all hypothesized to be a placebo. And the fun thing is also, all the metabolites found in Rafflesia is basically not very unique. They can be found almost 95 to 99% of the metabolites that currently listed in the paper can be found in other plants, domesticated plants which are not endangered. So we can use that instead from harvesting wildlife from Rafflesia, don't you think so? And so what is the future from this? Well, to unlock more about the behavior of Rafflesia, we have to do a lot of omics study relating to genomics, transcriptomics, perhaps metabolomics to see what's inside the Rafflesia, because Rafflesia basically has a very streamlined genome. We need more characterization of the gene for the metabolic pathway, and also required analysis for the plastid genome, because based on Molina et al. Study in 2014, the plastid of Rafflesia is basically doesn't seem to be quite similar to normal plastid. They have mysterious plastid-like thingy inside the cells, which I don't know what inside it. And perhaps we need to do metagenomic study to understand what microbial symbiosis growing with Rafflesia and tetrastigma that possibly become the answer for the mysterious reason behind the Rafflesia life cycle. Who knew? And then we need to do more developmental study to understand to unlock what happened in the vegetative life cycle of the Rafflesia. We want to do perhaps the in vitro culture. And by the way, I give you a free idea. Perhaps any of you want to do it in the future, because for now I want to do it more into the genomics level. So we have to do it all but together, because the study of Rafflesia cannot be done by Philippines. It has to be done with all of us, Indonesia, Malaysia, and we have to do collaboration with this. So this is my idea. We haven't done the liquid media tissue culture, host plant cultivation during tissue culture. And then we need to see the endophyte study across different Rafflesia species. We need to understand the behavior of the endophyte inside the different species, or inside of tetrastigma in the different species. And do you know that sapria, aside of growing in tetrastigma, it was told that it grows also in fetus. And then better sampling methods, because whenever we do the sampling using a histological analysis, it kills the entire population of the one single vine, right? Perhaps we can do something that that reduce the invasiveness and damage of sampling. Perhaps something modified from the computer tomography, magnetic sensing, something like that. We need to do that. And propagation optimization. And to do this, we have to collaborate with the people and local activists for this. We need public inclusivity, international collaboration, as I said before. And there are many claims. Do you know that in Bukit Tinggi and Sabah, someone has told to be succeed to grow Rafflesia from seed. And when I talked with Dr. Molina, I said like, okay, I think the key is how deep is the section is the cutting for the host, so the seed will grow perfectly. Perhaps the slicing, the sectioning, no, not the sectioning, the slicing incision has to be in the correct depth that allow the seed to reach the vascular cambium, hence to initiate the growth. And then there's a claim from a local gardener in Bengkulu that they managed to grow Rafflesia from infected host cuttings. Imagine that we do that on grafting, but this time is on cuttings. Okay. Now for the epilogue part, we need more study. That's one fundamental thing. We need more study. We need for improvement to make it better, and then we have to include the technology, the advance of biotechnology, bioinformatics, and other stuff to support our discovery, and perhaps to make it the study less invasive. And then we have to do it in a collaborative way. So more brains will be involved for this and to support the conservation program of Rafflesia. And we need to access better human practice with non-researcher, because I think there are no more than us, the local people, local gardener, activists who knew. The answer is out there and we just don't know it yet. So that's it from me. Thank you. I'm open to any question and any suggestion from you, all of you. Thank you.
Thank you, sir. Thank you very
You're
much.
welcome.
Okay, so at this point, we would like to let me share my screen. Let me see. I'll stop share. I will have to share my screen now so you can see. Okay. Thank you, Sir Wikaksono, for that wonderful talk. Okay, at this moment, I'm sure that there are already questions in our Slido link. Okay, I'm sure it's already flooded with lots of questions for our speakers for tonight. Okay, so without further ado, let me now turn you over to Dr. Joycelyn Humawan, our Q&A facilitator for our open forum.
Can you hear me loud and clear?
Yes.
Okay. Thank you, Dr. Bascos and Dr. Wikaksono. We had a treat with Rafflesia Developmental Biology tonight. I will proceed with the questions from our Slido. The first question is for Dr. Bascos. Anonymous. For Dr. Bascos, this is such a fascinating and engaging talk. Are Rafflesia plants the only plants that start from a pro-embryo?
I'm not sure if they're the only plants to start from a pro embryo, because there are other parasitic plants that are endophytic. So they start out just like Rafflesia, but the main difference is that for the other plants, they would have an external shoot. You can see them attached to the host. So perhaps, yeah, it's possible. I'm really not sure which specific plants would just have a pro embryo like Rafflesia, because it is the only plant that I know that really does not form any vegetative organs at any
All
point
right.
in its life. Yeah.
Okay. This question. This another question is for you, Dr. Erica. Um, For endophytes that are in rows, are there indications that they're slightly different? They are in slightly different developmental stages, say younger to older from one end to other. Again, I will read the question. For endophytes that are in rows, are there
In what?
indications that
In what?
yes,
In rows?
in rows, say younger to older or from one end to other.
Well, based on what we saw in all of those thousands of sections, so they really just start very near the vascular cambium. So in most sections, either they are in the vascular cambium or they are in the regions very near the vascular cambium. So we are assuming that those are the earlier stages as compared to the section showing that all of the endophyte cells are now scattered in the inner parts of the xylem and the outer parts of the phloem. So that's how we saw it in the sections.
Okay. Thank you, Erica. This other question is for Mr. Wicaksono. Sir, have there been any attempts to manually infect host stems with Rafflesia seeds or with Rafflesia endophytes to see if it can grow on stems?
Thank you for the question. You mean the stem cuttings and with the seeds intact, right? Or something like that?
Manually infecting host stems with seeds
So
or
far,
endophytes.
I see. Thank you. So far in my experience, it haven't been it hasn't been done for until now. All the experiment for the inoculation, it's inoculation or the introduction of the Rafflesia seeds to tetrastigma is often done in the root, but itself is also done in situ. You haven't tried in the ex situ by, for example, cutting the root and then doing it somewhere. Last time in 2020, we want to see the growth of the Rafflesia infected tetrastigma, both from the cuttings of roots and stems. Currently, it's still being in progress in Busofi, but well, let's see what happened in the next years. I don't know about that. It's still in progress,
Okay.
but no one's tried in on the seeds.
All right. This another question is for you, Mr. Wekaksono. This is from Jillian Uy from UP Diliman. For Mr. Wekaksono, in the 2019 study, is there a reason why Rafflesia patma does not have a phloem, or is it a characteristic of endophytes? Thank you.
In 2009 study, or you mean the one that I show the paper
2019
on the
study. In the 2019 study for Rafflesia patma.
No, I mean because the study that showed the vascular tissue of Rafflesia is my study in 2020. My study in 2019 is about the endophytes reveal on the tetrastigma. Or should I can I also answer for that as well?
Yes.
Okay.
Yes, po.
That's basically what I saw, and I don't know why, but for that the Rafflesia has not phloem at all. But from what I read from the reference, it is also quite rare to see parasitic plants to have a phloem. The only documented parasitic plant that has a phloem tissue is, if I remember correctly, is it either Pilostyles or Cytinus, either of these plants. But because the reason is because they don't have any photosynthetic organs, hence they only take the they only need to get the nutrients from the host, and that can be done directly from the xylem. And from the distribution itself, it will be handled from what I read, using the capability of the vascular parenchyma tissue.
All right. Thank you, sir. This question is for the two speakers. Okay, so what is the economic significance of studying Rafflesia in your respective countries? Again, what is the economic significance of studying Rafflesia in your respective countries? This is anonymous. This is an anonymous question. Who gets to answer first? Dr. Bascos?
Well, actually, that's something that's hard to answer because we're not trying to market Rafflesia as like for a drug source or anything like that, because most of them are threatened or critically endangered. So we're not trying to market them as potential sources of drugs or other compounds. But maybe in terms of tourism, because especially in the Philippines, like these plants are only found here. We have 13 species in total, so marami in our country, and then they're only found here. And I think it's very important that we do something to conserve, to protect these species that are only found here. And also, I think that these plants are very important kasi in terms of evolution, sila yung pinakaiba of all the plants. So we can really learn many things from this genus of extraordinary plants.
Yeah, I agree. What about Sir Wikaksono for the same question?
Well, basically my answer is quite the same. It will be good to support the tourism reason, and also because if we want to study, the study relating to the Rafflesia itself is not complete. There's a lot of mystery inside that, and by studying more Rafflesia, it will boost our understanding for any other plant related study. And also, in my opinion, we haven't known what is the direct impact of Rafflesia and the surrounding environment. Imagine if Rafflesia is removed from this ecosystem, it could lead to some disastrous problem in the environment. We don't know yet, but for what I know, every individual, every species in an ecosystem is correlated to one another, and that's probably what happened with Rafflesia and its host and surrounding plants and other.
All right. Okay, this next question is also for the two speakers. Are there any studies on the biological activities of compounds isolated from Rafflesia?
Yeah, I think Adito would be the best one to answer that.
Yes, Erica, go. Who gets to answer it first?
Do you want me to answer me first?
Sige po. Yes, Sir Wikaksono.
The study for the metabolites, well, so far from what I learned, Rafflesia has a broad range of metabolites from alkaloids, consists of nicotine and caffeine, and then there's a lot of phenolics, at least 5 gallotannins, and well, from what I study in using the molecular docking study, as I mentioned in my slide, these metabolites are basically good enough to support the fact that there's metabolite is good for anti-cholesterol and some other potential. But these metabolites are available also to other plants which are not endangered. So why should we harm Rafflesia to get these metabolites? That's my opinion. So well, and well, in the future, we have to study other metabolites from rafflesia, including the hormone or something that probably support us for the rafflesia conservation study in the future.
Okay. Probably one last question for the two speakers. This one question is for Dr. Bascos. Um, Considering Rafflesia consolidate is critically endangered in the Philippines, how many individuals were allowed for collection by DANR for your study?
Actually, we try to minimize the number of individuals from which we collected from, especially for the stem segments, because we were really very scared that we would kill all of these host individuals. But for the roots, since they are adventitious roots, so we can do cuttings. There's really no worry for the roots. But for the stems, we just sampled from 5 individuals for the stem part. But then again, we continuously monitored all of these vines in the area, and none of them died. So we are thinking that maybe the meristems there, the cells there are actively dividing. They are meristematic. That's why it's also very easy to do stem cuttings and then you plant them and they will just grow. So they're very active in terms of that. They are, meristematic activity. So I think that's one of the things that we can say, that we really did not kill any of those hosts in the area there.
Okay. Thank you, Erica. This one is for Mr. Wikaksono. Thank you for the interesting talk. What do you think is the main hindrance in the in vitro culture of Rafflesia sp?
Okay, the main problem here is basically when you do the sampling, you're making the explant for rafflesia, they tend to brown in the several hours, and that from what I learned in plant physiology, they will inhibit the growth and the enzymatic process for that lead to development. So we have to alleviate this problem related to browning, and to continuously induce the growth. And in the same time, this is the important part, to inhibit any potential contaminants, because I read a paper in 2011 by Refai et al. Stating that Rafflesia has endophytic fungi grow inside it. So basically, when you do the explant for the tissue culture, the fungi will grow alongside the sex plants. That's the problem.
All right. I think there are still several questions here in Slido, but we have to proceed with the next part of the program. So for those who are still interested in reaching out to our speakers, you can send them an email and direct your questions to the authors. Okay, so I think that wraps up our Q&A portion. So I'm turning you over to Ma'am Joanne. Thank you, everyone.
Okay, thank you very much, Dr. Joyce, for facilitating our Q&A portion of our session tonight. Okay, so at this juncture, we would like to show first our appreciation to our speakers. So this certificate of appreciation is being awarded to Erica Marie A. Bascos, PhD, for the generous sharing of knowledge and expertise as plenary speaker during the session 2 of the PSDB 13th Annual Scientific Conference held online on November 17 to 20, 2021. Signed by Dino T. Tordesillas, PhD, PSDB Secretary, and Larry Ann Ipulan Colette, PhD, our PSDB President. Thank you, Dr. Bascos. Thank you very much for gracing our webinar for tonight.
You're very welcome.
Okay. Likewise, we would like to award the Certificate of Appreciation to Mr. Adhitio Wicaksono for the genuine sharing of knowledge and expertise as resource speaker during session 2 of the PSDB 13th Annual Scientific Conference held online on November 17 to 20, 2021. Signed by Dino T. Tordesillas, PhD, PSDB Secretary, and Larry Ann Ipolan Colette, PhD, our PSDB President. Again, thank you very much. Thank you, Sir Wicaksono. Thank you very much for our speakers,
You're
Dr.
welcome.
Bascos and Sir Wicaksono. Thank you very much. We are indebted to you for sharing with us your expertise and also to our participants. I'm sure they have learned many things, especially about Rafflesia, a very interesting plant, should I say. Okay, so again, thank you very much. So we are now moving on to our next part of the program. So at
Ma'am
this
John,
point,
pa-share po ng screen. Sorry.
yeah, I am not sharing it. I'm sorry. Okay, wait. I can't seem to. Wait lang po. Yes, wait lang. I'm sorry. So hindi pala siya naka-share. Okay, so is it visible now?
Yes po, ma'am.
Okay, thank you very much. So I'm sorry for that. Okay, I forgot I was not sharing the screen. So this is our certificate for Dr. Erica Marie Bascos. And this is our certificate for Mr. Adhitio Wicaksono. Okay, again, thank you very much, our speakers. Okay, thank you for sharing us your expertise. Okay. So at this point, we would like to first invite our speakers and our participants to please turn on your cameras and wear your best smile for a photo op. Okay, so it will be Sir Jasheen and Sir Jordan, any of them. Please do the honors. And please do not leave yet as we have posters that we will be showing you. I think 3 posters. And we will also be announcing the winner for the photo taking contest. Okay, thank you very much.
Alright, paki-on po lahat ng camera natin. Wait lang po. Alright, yan. Meron po ulit 3 page, so pakitagalan na lang ulit yung smiles. Okay, we'll be taking the photo na po. In 3, 2, 1, smile. Another one, 1, 2, 3, smile. Next page. Okay, 1, 2, 3, smile. And the last page. 1, 2, 3, smile. Alright, thank you po.
Yay! Thank you very much. Thank you. Okay, so moving on to the next part of our program. Am I still sharing the screen?
Wala po ulit, ma'am.
Wala po ulit. O, sige po. Okay po, no problem. Okay, so again, so we are now on to our next part of the program. Okay, so again, participants, we have virtual poster presentations. Okay, we have them online. So I think the link is provided in the chat box. So if you have not seen any of them yet, so we are inviting you to please see them. But in the meantime, let us have a glimpse of three of those posters right now. Okay, they're just these are just short videos. Okay, so sige po. Everyone,
Wait lang po, mama.
yes po.
Share screen. Basic. Ayan. Share na po natin. Share sound. Alright.
Good day. I'm Alicia Perez, and presented in my poster is my undergraduate thesis entitled Assessment of the Teratogenic Effects of Cyclophosphamide on Danuario Embryonic Heart Development. Cyclophosphamide, or CPA, is a known alkylating agent anti-cancer drug. However, this drug is considered as a proteratogen since its bioactivation can lead to teratogenic effects on offspring. Previous studies on its teratogenicity in zebrafish focus on the overall effects in the early stages of its development. In this study, the teratogenic effects of CPA in the early and late stages of development of zebrafish embryo hearts were determined. Zebrafish Danuario teratogenicity assay with the metabolic activation system, more specifically the rat liver microsomes, was performed. Some of the findings include heart malformations, including enlarged ventricle and pericardial edema. This is during the early stages of development. As for the late stages of development, lethal and teratogenic effects such as enlarged atria and ventricles and decrease in the size of the ventricle relative to the heart were determined. Additionally, it was generally observed that increasing concentrations of CPA led to the increased enlargement of the heart chambers. The teratogenic effects that were observed was due to the presence of metabolic activation system, which bioactivated CPA, allowing its metabolites phosphoramide mustard and acrolein to produce an alkylating effect. As for the enlargement of the heart chambers, it can be due to either the presence of gene mutations, such as the GLAS and SANTA mutants, or alteration in heart functions. Lastly, the general trend observed is due to the lethal and teratogenic effects being concentration dependent. Please refer to the poster for more information. Thank you.
For our next one po,
Good day. I'm Alicia Perez.
mali, sorry.
Good day. I will be presenting a meta-analysis on the sex bias incidence of cleft palate and the laterality of unilateral cleft lip between males and females. Before I proceed, I would like to define the terms bias and laterality. In the context of the study, bias shall refer to the tendency towards a particular side, that is, males versus females, while laterality shall be synonymous to dexterity, which is left versus right. Clefts of the lip and palate are the fourth most common birth defects and have the highest prevalence among all congenital craniofacial deformities. The Philippines exhibits a significant birth occurrence of cleft lip and palate with a frequency of 1 in 500 live births, making oral clefting a critical health problem in the country. The study thus aimed to investigate the sex bias incidence of cleft palate and to assess the laterality bias of cleft lip along with probable factors causing such. Through an extensive systematic search and screening of various databases, execution of meta-analysis via MedCalc, and the quantification of publication bias through the generation of funnel plots. The results of the meta-analysis on the sex bias incidence of cleft palate revealed that there was a higher number of individuals with cleft palate in females compared to males, which generated an I-square statistic equal to 73.21%. One of the possible causative factors considered is that the female secondary palate closes later than that of a male of the same age. Through the meta-analysis on the unilateral cleft lip laterality in females, it was found that there was a higher prevalence of left unilateral cleft lip with an I-square statistic of 64.79%. However, publication bias was present as reported by the Eggers test p-value of 0.0294. The tendency towards the left side may be due to the difference in the vascular supply of the blood vessels on the left versus right side of the face as suggested by biological models. Although the same trend was observed for the results of the meta-analysis on the unilateral cleft lip laterality in males, it is not statistically confirmed due to the high level of heterogeneity detected among the studies, with an I-square statistic of 98.21%. It is thus recommended to conduct subgroup analysis to investigate heterogeneity in pooled effect estimates and to exhaust literature from all databases as much as possible. Since females exhibited a higher prevalence of cleft palate, hormonal manipulation using animal models may also be performed. For instance, estrogen levels may be increased, and the models may then be observed to see whether the tendency for cleft palate would increase as well. Thank you.
Then the last one.
Good morning. I'm Christine Padilla, and I'm here to present my poster on my thesis entitled Optimization of the Protocol for the Transplantation of MCF7 mammospheres on the duct chorioallantoic membrane. So breast cancer is the most common type of cancer affecting women, accounting for the majority of cancer deaths worldwide. Over the years, there have been tremendous developments in the detection and treatment of this disease, and for several decades, the MCF7 breast adenocarcinoma cell line has been the most extensively used both in vitro and in vivo in cancer biology studies. Despite the significant use of both approach, however, in vivo studies possess several advantages, and one of which is the capacity to observe the cancer under conditions resembling the natural microenvironment. The golden standard murine models are widely being used for tumor transplantation, metastasis detection, and anti-cancer activity assessments. With its increasing contribution to cancer research, however, come its high cost and, more importantly, the significant number of sacrificed animals. Therefore, it is invaluable at this point to develop alternative models which allow the conduct of the same, yet more humane animal studies. So here we present the chick embryo chorioallantoic membrane model, which takes advantage of the vascular network of this extraembryonic membrane. As a matter of fact, this cell line has already been proven to form tumors on the chick cam. Note, however, that considering the higher availability of fertilized duck eggs as compared to fertilized chicken eggs, here in the Philippines, we have decided to adapt the model to the former instead. So our results can be divided into 4 parts. So here we can see that we determined the human cancer cells and cancer types liable for in vivo implantation. Next, we then check the applicability of the duck egg to the transplantation of MCF7 cancer cells using 2 setups. So the first one involved a negative control group treated with PSP and an experimental group inoculated with MCF7-derived mammospheres. Here we have observed the emergence of histological alterations, namely cell infiltrations and dense mesenchyme, which are both suggestive of a possible tumor formation. For the second setup, we now decided to introduce MCF7 cancer cell suspension in the negative control and MCF7-derived mammospheres in the experimental group. So here we have seen the emergence of tumor nests, which are comparable to the ones observed in the study of Rebiti on pancreatic cancer cells. The third part of the study involved the markers which can possibly be used to detect the cancer cells transplanted on the cam. And lastly, we also present the uses, advantages, and disadvantages of the model. Thank you.
That's it po. Okay na po, Ma'am Joanne. Nakamute po kayo, Ma'am.
Okay. Okay, so I will share again my slide. Okay. Okay, so there you are, participants. You've already seen 3 of our poster presentations. So if you want to see more of them, you can click on the link that's given in the chat box, so you can view all the posters of our other PSDB enthusiasts. Okay, so next, okay, so and for the awaited part, okay, we will now be announcing the winner of our photo contest. Okay, I'll turn you over to our PSDB president, Dr. Larry Ipulan Colette. Ma'am?
Thank you, Ma'am Joanne. So I'm just gonna show you the different entries, some of the entries of our photo contest. Okay, so here are some of our entries from Miss Ria, from Sir Rodel, from one of our board members, si Ma'am Joyceline. And then we also have food platter from Bonita. And again, one from our board members, Sir Dino. Pagbaliktad ba nito, Rafflesia ba siya? So, and then, we have another from older millennial. And then from Clarice. And then from RJ, showcasing their plants. And we have a puppy participant in this photo from Doc J. So the winner of today's photo contest is Miss Ria Oblanca with the caption, "The beauty of Rafflesia is here." And she is also very beautiful in this picture. So congratulations, Miss Ria Oblanca. Our secretariat will contact you in Twitter to get your information and details for your prize. Thank you very much for all who submitted their photos in our Twitter and Facebook accounts.
Thank you very much, Doc Larry. Okay, so I agree, no? Maganda, our winner is also beautiful. She is beautiful, no? Our winner. Okay, so again, thank you very much for our participants, especially for those who sent in their photos to join in our photo contest. Okay, so before we end today's session, let me remind our participants to please do not forget to answer our session evaluation, okay, which will be open until 11:59 p.m., especially for those who would like to get a certificate of participation. Okay. And to formally close our session for the day, our second session, again, we, PSDB, in cooperation with Macare Medicals Incorporated, would like to thank our loyal sponsors, Merck, PhilAnaServe Incorporated, and MedTest. Thank you, sponsors, for your support. Also, we would like to thank our resource speaker, Sir Adhityo Wicasono and Dr. Erica Marie Bascos, for sharing with us your expertise. And of course, thank you for all our dear participants for joining us in today's session. We hope to see you again tomorrow for our third session. Same time, same place. Thank you very much. Good night. Thank you, everyone.
Thank you, everyone, and thank you for our speakers.
See you tomorrow.
Hello, Dr. Herrera.
Thank you so much.
Yes, Dr. Herrera, thank you.
Hello, Ma'am Bell.
Okay, thank you, everyone. Okay. Thank you,
Sir
sir.
Dino, Rafflesia ba yan pag binaliktad?
Sir Dano.
Nagagalit siya pag binabaliktad din.
Sir, pag nanalo po ba dun sa photo contest, pwedeng i-wave kasi board?
Baka daw, baka it's not, good practice.
Oo nga.
Hi, Dr. Herrera! Merry Christmas!
Thank you very much po, everyone.
Ma'am, nakamute po kayo, Ma'am Herrera. We can't hear you po.
Ayan.
Hi, ma'am.
Hi,
Good
ma'am.
evening. Thank you po for
See
attending.
all of you.
Yes, ma'am.
Happy to see you, Ma'am Bell. Bye. Nako si Joy. Miss you.
How
Tapos
about
ko na
Ma'am
po yung
Sonia?
recording eh.
Yes,
Okay,
please.
thank you.
Thank you to our participants. Tomorrow po
Thank you.
ulit.
Same time, same place.